KAP Corp presents the process of self-signaling in tyrosine kinase receptors.
Here we have two tyrosine kinase receptors. For simplicity purposes, we will call them Phil and Susan. Phil and Susan do not like each other as of right now. Maybe Phil forgot Susan's birthday. Both of these receptors have a high affinity for certain ligand. This ligand correlates to the shape of the receptor. We can think of this ligand as a credit card. Phil receives his new credit card in his mailbox and tells Susan he got paid. These two are now happy again and give each other a hug, which they call a dimer. The tyrosine kinase proteins are now phosphorylized. We can relate this to how Phil and Susan start buying a whole bunch of things because they think they have a lot of money. The phosphate is then used to activate protein in transduction. The activated proteins go through a series of reactions leading to a response. In reality, Phil only works at Burger King and has no money. The bank starts to take things they just bought and gives them to their own family. The bankers' families are now happy. Eventually, the credit card company revokes Phil's credit card and Susan gets angry and pushes Phil away. This is when the ligand detaches from the receptor and the transduction process stops.
A real example of this process is through insulin, which is a ligand. The insulin attaches to the receptors of the tyrosine kinase and creates a dimer. As relay proteins take phosphates from the tyrosine kinase, the process of transduction begins. The active relay proteins activate the first kinase protein. This active protein activates other proteins later in the process. This is called a phosphorylation cascade. Amplification can occur during this process. The phosphorylation cascade eventually leads to a cell response, usually an indication to begin gene transcription. An example of this cell response can also be seen from the insulin example. A pathway of phosphorylation process follows is the PL3K signaling pathway. The end of this pathway sends GLUT4 to the cell membrane to allow glucose to enter the cell to be converted to ATP or glycogen. An organ that uses this receptor is the liver, as it takes in glucose to make glycogen.
Some examples of diseases caused by a failure of the tyrosine kinase receptor are type 1 diabetes and many types of cancer. In type 1 diabetes, the body cannot produce insulin properly, and thus the ligands that would typically attach to the receptor cannot be used to break down glucose. Instead, a person with type 1 diabetes must inject insulin into their bloodstream in order to elicit the response previously mentioned. The epidermal growth factor receptor is integral in the formation of many cancers, such as pancreatic cancer. Growth factors bind to these receptors and produce a cascade of signaling events that culminate in the growth of malignant tumors and the development of tumors outside of the original cancer site. And that's KAP Corp, bringing you the gist of tyrosine kinase receptors.